Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we'll talk about inhibitors and uncouplers of the electron transport chain oxidative phosphorylation pathway. In the previous video, we talked about genetic defects in the electron transport chain oxidative phosphorylation. Today, we will look at chemicals which inhibit the electron transport chain, chemicals that inhibit ATP synthase, and chemicals that uncouple electron transport chain from oxidative phosphorylation, what we call uncouplers. Just a quick reminder, as we discussed in previous videos, we have two parts. So we have electron transfer. That's how electrons from NADH or FADH2 are transferred to oxygen. So this part here is electron transfer. Since oxygen, most of your oxygen in your cells is used up here, in a way, oxygen consumption, if we monitor oxygen consumption, we are getting an idea of how fast this electron transfer is happening. The second part is ATP synthesis or oxidative phosphorylation. So that happens here by ATP synthase. This is the enzyme that takes ATP and PI and generates ATP. And according to the chemiosmotic theory, these two are coupled. ETC is coupled, and that's what the slash here means. ETC is coupled to oxidative phosphorylation. There are several chemicals that can inhibit the electron transport chain as well as oxidative phosphorylation. We can classify these chemicals either as electron chain inhibitors, ATP synthase inhibitors, or uncouplers. Remember that complex 1, 2, 3, 4, as well as ATP synthase, these are all enzymes, therefore they can be inhibited by chemicals. Here are a few examples of these chemicals that can inhibit electron transport chain oxidative phosphorylation. Complex 1 can be inhibited by rotenone, that's a fish poison as well as an insecticide. It can be inhibited by amytal, which is a sedative, CNS depressant, and complex 1 can also be inhibited by cocaine. So rotenone, amytal, cocaine are all examples of complex 1 inhibitors. Recall that the enzyme succinate dehydrogenase of the Krebs cycle is closely associated with complex 2. Malonate is a competitive inhibitor of succinate dehydrogenase and therefore an inhibitor of complex 2. The antibiotic antimycin A, which is also used as a fish poison to get rid of invasive fish species, is an inhibitor of complex 3. Azide, that is often used as a preservative, carbon monoxide, cyanide salts, as well as methamphetamines, these are all examples of chemicals that will inhibit electron transfer in complex 4. So besides the ETC inhibitors, we also have the ATP synthase inhibitors, chemicals that inhibit this enzyme ATP synthase. And examples are the antibiotics oligomycin, ephrapektins, orovertins, as well as the synthetic chemical DCCD. And finally, we have the uncouplers. These are chemicals such as dinitrophenol and salicylate. So these are chemicals that uncouple oxidative phosphorylation from the electron transport chain. Let's see how these inhibitors as well as uncouplers have been used to understand the coupling between electron transport chain with oxidative phosphorylation. So in these experiments, we have isolated mitochondria, and these are incubated in a container, and there is an oxygen electrode that can monitor the amount of oxygen in the container. Since oxygen is the final acceptor of electrons in the electron transport chain, we can conveniently measure the activity of the electron transport chain by monitoring the oxygen consumption by the mitochondria, and that is done by the oxygen electrode. The activity of ATP synthase can be conveniently measured by measuring the concentration of ATP that is produced by this enzyme. And the concentration of ATP can be measured by several different methods, including chemiluminescence, as well as by using radioisotopes such as P32. So in these experiments, basically what we do is we measure, let's say, the concentration here of ATP or the concentration of oxygen by the oxygen electrode with time. So let's see what happens when we add certain chemicals to this system, to the mitochondria, and see what happens to their PO2 as well as to the concentration of ATP. So we have the baseline oxygen concentration here, PO2. Now let's say we add glucose to the container and look at the baseline concentration, and the PO2 remains the same, and that's because glucose cannot be metabolized by the mitochondria. So since glucose cannot be metabolized by mitochondria, the PO2 levels do not change. In the next step, let's say we add ADP and PI. Remember, ADP and PI are the substrates for ATP synthase. And we monitor again the PO2. Once again, although we give ADP and PI, the concentration of PO2 doesn't change. So basically, there's no electron transport chain happening here, and therefore no ATP that is synthesized during this time. Now, once we add a fuel such as citrate can be metabolized by the mitochondria, and now we see the electron transport chain happening, and oxygen is being consumed as the ETC activity occurs. And since the ETC activity is occurring and there is ADP and PI present, we also see ATP synthesis going up. Now let's say we add oligomycin. Now oligomycin is an inhibitor of ATP synthase, so not surprising, ATP synthesis stops. You can see ATP synthesis stops here. And interestingly, not only does ATP synthesis stop, electron transfer stops, the ETC activity stops. So this clearly proves that ATP synthesis is coupled to electron transfer. Okay, so this is a very nice experiment to show the chemiosmotic theory. ATP synthesis is coupled to the electron transport chain. There is still no ETC activity as shown by the lack of oxygen consumption, even by adding more fuel such as succinate, and that's because still the ATP synthase is blocked by oligomycin, and ATP synthase is coupled to electron transport chain. However, now if you add an uncoupler such as dinitrophenol, now we see the OP being uncoupled from the ETC. So this will cause electron transport chain to occur. So electron transfer will occur. However, ATP synthesis is still stopped. Now that's because not only of oligomycin, but also because of the uncoupling. If we now add rotenin, which is the inhibitor for complex 1 here, so it inhibits complex 1, we see that the rate of oxygen consumption slows down a little bit compared to previous. And that's because complex 1 is inhibited. However, succinate enters through complex 2. So that will still allow electron transfer to occur in the electron transport chain, but in this case between succinate and oxygen. ATP synthesis, however, is still doesn't happen. That's because, once again, inhibition by oligomycin, and there is an uncoupler DNP present. Now, if we add cyanide, cyanide is an inhibitor of complex 4. So in this case, neither electrons from NADH or FADH2 can be accepted by oxygen, and electron transport chain activity completely comes to a halt. ATP synthesis still does not happen, now not only because it is inhibited by oligomycin, there is an uncoupling between ETC and oxidative phosphorylation, and now there is also no electron transport that is happening, and without electron transport, ATP synthesis will not happen since the two are coupled. So what do you think the graphs would look like if you added these chemicals in this order here: glucose, ADP, PI, citrate, and cyanide? You can pause the video now and then try it yourself, and we'll look at the answer in a few seconds. So as before, once we add citrate, which is a fuel, oxygen will be consumed, the electron transport chain will happen, and since the electron transport chain is happening, there is ADP PI, so ATP will be synthesized. However, once we add cyanide, cyanide will inhibit complex 4, that will completely stop the electron transfer, either from NADH or FADH2, doesn't matter, because cyanide inhibits complex 4. And if electron transport chain doesn't happen, there's no oxygen consumption, ATP synthesis will also stop because these two are coupled. Here's another one to try, see what kind of graphs you get when you add these chemicals in the following order. So once again, as you add citrate, oxygen will be consumed, ATP will be synthesized. Now if you add the uncoupler DNP, the electron transport chain will continue. So that's not blocked, right? Because citrate is a fuel and it's going to be sending electrons to oxygen. So this will keep on happening, but because of the uncoupler, these two are no more coupled. ETC is no more coupled to oxidative phosphorylation, so ATP synthesis stops. So there is no ATP synthesis here. That's the uncoupling. So hopefully you would appreciate the coupling that happens between the electron transport chain and oxidative phosphorylation. Now, how do these uncoupling chemicals such as dinitrophenol work? Uncouplers are basically small hydrophobic chemicals which have a functional group that can either exist as the protonated form or the unprotonated form around this pH 7 to 8. This is the pH difference between across the inner mitochondrial membrane. So one of the best examples of an uncoupler is dinitrophenol. So you can see this is basically a small, very hydrophobic group, and there is this hydroxyl, this phenol hydroxyl group that has a pK around this area. So an uncoupler such as DNP at a lower pH, such as pH 7, will be mostly in its protonated form. The OH group will be mostly in a protonated form. Since the uncoupler is hydrophobic and small, it can cross the inner mitochondrial membrane. Once it goes to the matrix site where the pH is higher, it will tend to lose a lot of these protons to become unprotonated, the unprotonated form O-. This unprotonated form will then again cross the inner mitochondrial membrane, get out to the other side, and there it will pick up a proton, another proton, and become protonated. And this once again goes inside, and so this cycle repeats, thereby bringing protons from this side of the inner mitochondrial membrane into the matrix site. So in the presence of an uncoupler, instead of the protons going through ATP synthase, the protons are going through the matrix by the uncoupling agent. Here's a case reported in literature about, there are several cases of DNP poisoning, but here's one example. So there's a 25-year-old male, was a bodybuilder, ingested about 620 milligrams of dinitrophenol, and he was admitted to the hospital 4 hours after ingestion of dinitrophenol. The clinicians found him agitated. He had very high heart rate, tachycardia, about 170 beats per minute, normally 60 to 100. Very high body temperature. Instead of 37 Celsius, his was close to 39. High blood pressure, very high respiratory rate, about 41 instead of 12 to 16. He was sweating profusely. So because of the high body temperature, he had metabolic acidosis, so his blood pH was less than 7.4 on the acidic side. And his lactate concentration was elevated, 2.2 instead of a normal of less than 2. And he died 10 hours after DNP ingestion. So what happened? Well, the uncoupler, the DNP, as mentioned earlier, brings protons into the matrix and uncouples ATP synthase from electron transfer. So the energy, the electrochemical gradient energy, instead of being used for making ATP, it is released as heat. So basically a lot of heat it will be generated. Because of the uncoupling, ATP production in his body decreased. So the body compensates by sending more fuel to the electron transport chain. This will increase the electron transfer activity, but since it's still uncoupled, the energies will simply be released as heat. The body temperature increases, ATP production is still decreased, and to meet the energy demands, the body increases glycolysis. And as we mentioned earlier, for glycolysis to keep on happening, the pyruvate gets converted to lactate, and that increases the lactic acid concentration, leading to lactic acidosis, which results in a low blood pH, lower than 7.4. There is a lot of interest about uncoupling chemicals, because by uncoupling, basically what's happening is that all the food that you're eating, instead of being converted to ATP, it is simply coming out as heat. So this makes it a very good weight loss chemical, anti-obesity pill. So that's one reason why there's a lot of interest in finding a safe uncoupling chemical for users in obesity, for metabolic associated fatty liver disease, as well as for type 2 diabetes. So DNP is very dangerous because it has a very narrow therapeutic window. One can get into the toxicity range very easily. So there is a lot of research going on in finding modified DNP derivatives that can be used, which have a much safer profile. There are also some natural uncouplers. There is a protein called uncoupling protein. For example, UCP1 is an uncoupling protein. This protein is also found in the inner mitochondrial membrane, and what it does, it basically brings protons into the matrix, and that leads to generation of heat. This protein is found a lot in what we call brown fat. Okay, so we have white fat, which is adipocytes, fat cells that store fuel, store triglycerides. But in brown fat, white fat doesn't have much mitochondria. Brown fat has a lot of mitochondria. So the function of brown fat is not to store triglycerides. Instead, it is to generate heat, because for newborns, it's very important to keep them warm. Since their muscles are not very well developed, they cannot shiver very well to release heat. So the presence of brown fat that uncouples oxidative phosphorylation from ETC provides them with heat to keep them warm. So this is called non-shivering. Okay, it's not from muscles. Shivering comes from the action of muscles. Non-shivering thermogenesis. The thyroid hormone T3 actually increases the amount of UCP in brown fat, and that leads to more non-shivering thermogenesis. So people with higher thyroid hormones, hyperthyroid, they tend to feel warm most of the times, while individuals who have low thyroid hormones, hypothyroidism, they tend to feel colder. So that is all for now. Until the next video, have a wonderful day.